İnniki çıkışımız Neuroscience'da doktoranturasını edip görün Gulhumay Gardashova. Gulhumay Gardashova 2013 yıldan 2017 yıla çenni moleküler biyoloji uğrundan derecetnalan 2017 yılda University of Kansas Üniversitesi'nden Lawrence'daki ondan beri olsa UT Southwestern Dallas'taki UT Southwestern'a PhD'sini edip bör. Oraya işleyen işi neuroscience, rotayda asistanlığı diğer türlü publication'ları var, çap eden makaleleri var. Bununki teması İngiliz dilinde learning from fruit fly learning. Meyvesinin öğrenişinden ne öğrenip bilir diyen tema olan çıkış ediyor. Gulhumay Gardashova, floor is yours.
Thank you for that introduction, and thank you for inviting me to give a talk. My name is Gulhumay, and I'm a PhD student at UT Southwestern. And today I'm going to talk to you about what we can learn from studying how fruit flies learn. So we all came here to the symposium to learn new things. And in our lab, we study how your brain cells or neurons help you do that. More specifically, we're interested in how neurons help us learn and store information. And the first step to learn something new is to know that what we learned is new. So we're very sensitive to novelty, and whenever we detect something new, it evokes automatic attentional response. For example, this morning I saw a lot of new faces, and I decided to introduce myself to some. So this is called novelty detection. What is interesting about novelty detection is that as soon as we detect as something new, it becomes familiar, what we call transition to familiarity. For example, the new faces that I met earlier this morning are not new anymore, they're familiar. And this learning that occurs, that mediates this transition to familiarity, will prevent me from embarrassing myself by introducing myself to the same people again later today. So this novelty detection and transition to familiarity is almost everywhere. For example, I'll give you a very simple example with this dog. So in this video, you'll hear the same sound twice. And you will see how the dog responds to the sound. So the first time the sound is presented, the sound is novel, so the dog will turn and look away. The second time the sound is presented, it's novel, so the dog ignores it. So I'll play that. So you can't hear the sound.
Yes, we can.
Oh, okay. Okay, so anyway, so the sound is presented the first time the dog looks. You saw how the dog reacted to that sound. It looks to the side. Let me play that. So there's supposed to be a sound there, and then the dog looks. And then the same sound is presented the second time, but the dog just ignores it. So how do our brains, well, in this case, the dog's brain, decide to respond to this sound the first time, but not the second time? So before I go into explain you how that might work, I first want to go over how neural circuits work in general. So our sensory neurons detect sensor information. It can be vision, smell, or hearing. These sensory neurons send information to the brain, where this information is processed and the decision-making occurs. Then these interneurons send information to the motor neurons that carry out the behavior. So in the example that I gave you with the dog hears the novel sound, which activates the sensory neurons in the ear. These sensory neurons send information to the neurons in the brain, where this information is processed. And since the sound is novel, it decides to respond to that sound by changing its behavior, by alerting to that sound. However, when the sound is familiar, although the volume of the sound is the same and the sensory neurons and the sensory part of the brain that respond to the sound is the same, somehow this dog does not respond. And we think that this is because the connections between these interneurons and motor neurons are modified, or connections or synapses, and we call this synaptic plasticity, which is known to underlie learning. So this is this is our hypothesis of how venality detection and transition to familiarity might occur, but we actually don't know what are the neural circuits that underlie this learning. And in our lab, for this project, I'm interested in identifying these neural circuits and examining how they work. So what do we need to study how neural circuits work? First, we need the knowledge of the wiring diagram. What I mean by that is we need to know how one neuron connects to other neurons. We need to have a genetic handle for each neuron that will help us to manipulate the neural activity of these neurons and also record the neural activity from these neurons. I'll explain these tools in detail in later slides, but all of these requirements can be met by using Drosophila or melanogaster or fruit flies as a model system. So this system is an ideal system to study how neural circuits work because we have the connectome data. And what I mean by that is all of the neural connections that are in the fly brain are already mapped out. So we have the entire wiring diagram of the fly brain. In addition to that, we have a large collection of genetic drivers that help us label individual neuron types. So this permits us to use modern neuroscience techniques to study the neural circuits. And I'm going to go over some of the techniques that we use in the lab. And the first one is optogenetics. So in optogenetics, we can express a molecule, light-sensitive molecule, which is called channel rhodopsin. You don't see my pointer. We express this molecule called channel rhodopsin, which is light sensitive. So we can express this channel rhodopsin in any of the neurons of interest. And when we shine light on those neurons, the neurons get activated. So I'll give you an example. So in this study, the scientists decided to activate neurons involved in fighting and aggression. So here the flies are moving around, but when they shine light on these flies, they start fighting. So I can show you examples here. This fly is attacking that one. So you can see the flies fighting here. And you will see increases in the number of flies in each of the square. Basically, they're trying to fight each other. So by simply shining light on these neurons, we can activate their aggression neurons and they will fight. We can also inhibit the neurons using these optogenetics tools. Additionally, we can record neural activity from the neurons of interest. And we use a tool called calcium imaging. So we can express a molecule called GCAMP in the neurons of interest. So under normal conditions, when the neuron is silent, the GCAMP is very dim. But when the neurons get activated, this GCAMP senses that activity and it increases its brightness. It basically fluoresces. And we can use this change in fluorescence as a proxy for neural activity. And I'll give you an example here where we are, so this is a fly and it has a microscope on top of its head and we're recording the neural activity of these dopamine neurons that are sensitive to locomotion. So whenever the fly moves, you will see an uptick of the neural activity. And here are the neural activity traces. I'll show you. So here you see an uptick of neural activity and that will correlate with a behavior. So basically we can study the neural activity of any neurons of interest. Last but not least, modern advances in technology allow us to develop more precise behavioral assays, such as that use virtual reality. So here, the fly is in a bowl and it has a screen in front of it. So it's a virtual reality screen where fly is navigating in this virtual cone forest. These kind of tools allow us to study behaviors in a more precise manner. All of this make Drosophila model a playground for the systems neuroscientists where we can study neural circuits in a deeply manner. Okay, so this is flies, but do flies have similar behaviors as dogs or us? The answer is yes. So here we developed simple assay where flies are put in a constant state of grooming by dusting them with fine powder. So here when and we look at their behavioral responses by presenting different odors. So here we presented a novel odor to a fly. I'll show you the video in a second. So the fly initially grooms, and whenever we present the novel odor, it stops grooming and it's like alerts and starts moving around. So let me play this video. So it's grooming, and when the novel odor is presented, kind of this alerting behavior, and it starts moving around, which we call alerting behavior. But when we present familiar odor, when the odor is presented, it does not do anything. It just keeps grooming. And here's the quantification. So when the odor is presented the first time, when the odor is new, many of the flies do this alerting behavior, but with repeated presentations of the same odor, you don't see that alerting behavior. So it's basically similar to dog and sound. It's just fly and smell. So we wondered, what are the neural circuits that is involved in this novelty detection and transition to familiarity? Sorry. And I guess since we know, is she okay? Okay. Anyway, so we are interested in the neural circuits that are involved in this novelty and transition to familiarity. So since we know all the neurons in the fly brain, we decided to look for neurons that have differential activity in response to novel versus familiar odor. And we identified this neuron called MBON alpha prime 3, shown here. And I'm showing you the neural activity of these MBON in response to the odor. Let me pull up the pointer again. So here, this represents the odor whenever the odor is presented, and the top traces indicate the MBON activity, neural activity. So when the odor is presented the first time, you see the strong uptick of the neural activity, suggesting that this MBON alpha prime 3 is activated by the novel odor. But with repeated presentations, with a second presentation, the response is lower, and with repeated presentations, the neural activity response is gone altogether. And this is basically shown here. When the odor is new, there's high activity in this MBON alpha prime 3, and with the repeated presentations, that activity is lowered and gone. So if we look at the behavioral response and then the MBON activity, they're similar, suggesting that this MBON activity might be reason for the alerting behavior. And using this neuroscience tools, we can test that hypothesis. So first, we silence these MBON alpha prime 3. Basically, we inhibit it so they're no longer active. So in the control flies, when we present novel odor, they do this alerting behavior. They react to that novel odor. But when we silence this MBON alpha prime 3, the flies no longer respond to this novel odor, suggesting that these neurons are necessary for this alerting behavior. We did the opposite, where we activated these MBON alpha prime 3 neurons, and you can see increase in the alerting behavior in response to novel odor in comparison to the controls. And they do this alerting behavior even in the absence of the odor, suggesting this MBON alpha prime 3 is necessary and causal for this alerting behavior. So far, we have identified this MBON alpha prime 3 neurons that are neural correlates of novelty and familiarity. So when the novel odor is presented, the MBON alpha prime 3 is activated, which causes the alerting behavior. When the odor is familiar, the MBON alpha prime 3 is suppressed and there's no alerting behavior. So what is resulting in the suppression of MBON alpha prime 3 response? In other words, what is mediating this learning that accompanies this transition from novelty to familiarity? Well, from the literature, we know that dopamine system is involved in learning. It's involved in reward learning, punishment learning. And so we decided to look, since we know all the neurons in the brain, we decided to look whether dopaminergic neurons are connected to this MBON alpha prime 3 neurons. And we found this DAN alpha prime 3. DAN stands for dopaminergic neuron. And we decided to test, okay, are these dopaminergic Dan alpha prime 3 neurons is what's causing the suppression of M-bond response when presented with the familiar odors. So again, we tested that by first inhibiting these dopaminergic neurons. So here, the white and gray are controls, and we recorded from the M-bond neurons, we recorded their neural activity. So when presented with novel odor, control flies have high response to the novel odor, but it goes away. But when we silence these dopaminergic neurons, there's no reduction in the M-bond neural activity, suggesting that dopaminergic neurons are necessary for the suppression of M-bond response. We tested the opposite of that. We activated these dopaminergic neurons. And so if you compare to the controls, now the M-bond response is suppressed significantly, suggesting that this dopaminergic neurons are what mediating the M-bond suppression. So in summary, we have identified a simple circuit in the fly brain that underlies novelty detection and transition to familiarity. And how this occurs is when the flies are presented with novel odor, M-bond alpha prime 3 is activated and it causes alerting behavior. But when the familiar odor is presented, the M-bond response is suppressed, so there's no alerting behavior. And we found that this is mediated by dopaminergic neurons, where these dopaminergic neurons change the connection or synapses between these odor neurons and behavior neurons, which is synaptic plasticity. And this dopaminergic neurons are basically they're behind the learning that accompanies the transition from novelty to familiarity. Okay, you might ask, okay, these are just flies. Do these findings have any relevance in what might happen in mammalian brains such as ours? So despite the great difference in the complexity between flies and humans, so the flies have around 10 to the 5 neurons, and then humans have billions of neurons. Interestingly, the circuit organization is quite similar between flies and the humans. So here I'm showing you the circuit organization in the olfactory system, in the visual system, in the dopamine system that I just mentioned. So for example, the dopamine not only have the same function in the flies and humans, but it also how the neurons are organized in the brain are quite similar. So this common architecture might be a way of optimizing the circuits through evolutionary time to solve similar problems across different species. And we hope that what we find in the fly brains can be applied to what might happen in the human brains. And with that, I want to thank my mentor, Daisuke Hattori, and everyone in my lab. And if you're interested, the study was published, so you can go and read more about our study. And with that, I'd like to take any questions.
How does aging affect the learning capability of fruit flies?
So that's an interesting question. And I am not familiar with any studies that have addressed that, but we know that, so in the aging brains, the synapses between the neurons are deteriorated. So that's why we have memory decays in humans, and the similar processes occur in the fly brain. So I think the synapses between the neurons might be deteriorating over time, and that's my assumption. But I'm not familiar with any studies that have addressed that. Yeah, but I can get back to that person.
Okay. We have a question here.
Hello, I really liked your presentation. Good job on that.
Give me a second. We good? All right.
Okay, I was just curious, so the gene that is responsible for the alerting behavior, is it, are there any instances where there are people who lack this gene? Okay, who lack this gene and they basically don't have alerting behavior or like have issues with that?
So that's an interesting question.
With mutations in that gene?
Yeah. So what we found is actually a neuron. So the neurons have a lot of genes, and the dopamine is very important to mediate any behavior. So like dopamine is involved in movement behavior. So I think if we mutate dopamine itself, it might be lethal. But so what we have identified is the neuron. I'm not sure whether there are any, like, are you asking about the dopamine gene?
Another one, the alpha prime.
Yeah, it's a neuron. So I think there might be some conditions that make this circuit not as efficient. For example, I guess in Parkinson's disease, we know that Parkinson's disease is due to deterioration of these dopamine neurons, and we see deleterious effects with the movement, so they cannot initiate the movement. So I'm sure, like, if we like change something in the system, it might affect different things. Yeah.
Right. We have a question from Arara, Dr. Tomaji. Now, impressive work. Wondering if iPSC technology would be helpful in vitro manipulation of various sensory neurons.
So the question, let me read it. So the question is whether iPSC technology would be helpful in vitro manipulation of various sensory systems. I think, yeah, that's, so the question is whether iPSC technology would be helpful in vitro manipulation of various sensors. So iPSC technology is a technology where we can take these cells and we can turn them into any type of cell that we want. We can make these like small cells. We can make them neurons out of it. We can engineer different organs if we want that. So the question is, I think we can address many questions using iPSC technology. But of course, using iPSC technology is not the same as using an entire organism with its different types of cells. So in iPSC cell, it might be restricted to like 1 to 2 types of cells, but in the fly brain, there are numerous types of cells that that are all important to carry out the behaviors.
To conduct this study, how many flies you used?
A lot. I mean, I don't know. We don't even count them. Yeah.
I have some at home if you need some more.
Oh, that'll be great. Yeah.
Any other questions?
Yes.
You need to control.
That's turned on or? Okay, so my question is if the sensory behavior sensitivity actually varies by the certain species in terms of what kind of sensory we're talking about. So for instance, for dogs, for instance, sound might be a very, sensitive sensory channel, whereas for flies, it could be otters or something like that.
Yeah, for sure. I think for example, for flies, the smell is very important. Say they navigate through the environment using mostly their olfactory system. And for example, they don't have the same, I guess, hearing system as we humans do. So they actually do their hearing through their antennas that also detect odor. And I think for flies, it's most importantly odor, like smelling, and then comes vision. Yeah, does that answer your question? Yeah, and that's the reason why in our lab, we mostly focus on odor because like the flies have very, yeah.
So I was more interested in similar species, like for instance, among the mammals, like for humans and dogs or cows, monkeys. Because obviously there will be certain differences between flies and different plants, but in terms of like similarities, if we look at this similar species.
So I think for sure that, well, I'm not sure about the dogs, but for example, neuroscientists use mouse, right? So some mice they specifically use their odor system, or some mice they evolved to use different, like for example, the mice that live in the desert might have evolved their different sensory systems to kind of navigate through this world. So I think for sure there are differences between species.
Right. We have a question from Instagram. Do you track animal body parts? If so, Is it auto tracking or manual? Which tools and do you use?
Yeah. So the question is whether we track different parts of the animal. So I guess I can, is my presentation up? Yes,
Yeah.
I think so. So I guess first I can answer. It depends. I think the tools that are used to detect different parts are somewhat not to the level that we want. So we have used these tools that detect different parts, but these tools make mistakes. So we have to go back and re-annotate those videos. So in our lab, we use both, but we trust our like annotations, manual annotations in comparison to using the tools. But for example, in the, let me go back to the presentation. In one of the videos that I showed.
You need to share the.
Oh, I see. Never mind then.
Right.
Yeah, never mind.
Right. Zoom'dan sonra, can the activity of M-band synapses be modulated or modified by external factors or environmental stimuli? So can the activity of M-band synapses be modulated or modified by external factors or environment? So the answer is yes. The dopaminergic neurons that modify this M-band synapses, they actually respond to odors as well. Or these dopaminergic neurons respond to like shock or sugar. So these dopaminergic neurons receive information from the environment and they can modify the synapses. And there are not only dopaminergic neurons, but other types of neurons like serotonergic neurons, basically different types of neurons that innervate these synapses, and they can also modify the synapses. Yeah. I need to control the sound.
Yeah.
I will ask in English.
Yeah.
So thank you for the presentation. I liked it a lot. My questions is more like about the future. So if there is any path forward, any future researches about this, like a continuation of it, what would it be and what's the plan and when would it be?
Yeah, I think this story is somewhat complete. We published this study already, but I mean, for sure, we can, like, this is learning, right? And we can keep studying how learning occurs in different environments. Yeah, so right now, this is the completed project. Right now, I'm working on different projects, so I can tell you more about it. Like, for example, right now we're learning about where social information is stored. So we know that social experiences affect our behavior. We know that social isolation leads to depression, or social enrichment makes us happy. So, which suggests that we store social information somewhere in our brain, and we're trying to identify the circuits that store that social information and like how those circuits modify our behaviors. Yeah, this is somewhat connected to learning as well. Yeah, thank you.
Online, are there any known differences or variations in M-band synapses among different fruit fly strains or species?
So, are there any known differences or variations in M-band synapses among different fruit fly strains or species? I don't know the answer to that question. I think in the field, many labs use fruit flies, but there are labs who are like, who use other strains as well, but not as much. And yeah, I don't know that, but I can get back to you. But the mushroom body, the organization of the neurons, how the odor is received in the brain and then how that's processed is similar in many of the insect species. So I would assume, although it might not be exactly M-band alpha prime 3, it might be similar neuron, but the architecture or how the neurons work is, I think, similar across different insect species. Yeah.
Başka soru var mı?
Yes.
Maybe you already mentioned in the presentation. I might I might have missed it, but I'm curious about the mechanism that determines the null information versus the familiar information. You talked about the circuits and all of that, but if you can just add a little note more and elaborate how that works.
Okay, so I think I will have to show the circuit.
Go to
I'll
Zoom.
share, okay. Share screen. So what I didn't show you here is that this odor can activate both the odor sensor neurons, which are shown here in green, but they can also activate the dopaminergic neurons. So both the sensor neurons and the dopaminergic neurons are receiving kind of the same odor information. And whenever this dopaminergic neurons get activated, it kind of depresses the synapses between the like odor cells and the M bonds. Does that answer your question?
Way too technical for me, but somewhat, yes.
Sorry. You can ask me questions. I'll love to answer.
All right. What was the place where you were most patient in the lab?
What's the place? Okay. Since I have the
It really
slide.
takes a lot of patience to conduct these studies.
Yes. Since I have the slides up, I'll go back to the neural activity recording. So I think the most challenging, the question was, I guess, what was the hardest part of doing these experiments? I think this is the hardest experiment of all. So here the fly has a huge microscope and we're trying to detect how single neuron, the activity of single neuron changes. And it's very hard. So we have to, I don't know if you've seen fruit flies. So those are the flies that fly around the fruits. They're very tiny. So we have to like do a surgery on the fly brain to be able to put the microscope on top, which is very challenging and it takes lots of trials and errors.
It's impressive. Başka soru? Yok. Okay.
Yeah, thank you so much for listening.